Here we're going to talk about medications that are used to treat infectious disease. Basically what we'll do is take a look at different classes of antimicrobial drugs, and we'll talk about how those drugs work. One thing that all of these antimicrobial drugs have in common, at least to some extent, is that they all exhibit something called selective toxicity. This is a really important thing to have if you're going to develop a drug that is going to be used by people, because selectively toxic in this case just means that the drug is more harmful to the pathogen, like the bacterium or the virus, than it is to our cells. So again, all of the drugs that we're going to take a look at, they all exhibit some sort of selective toxicity. Otherwise, we wouldn't use them because they would harm us. Before we take a look at how antimicrobial drugs work, we'll first take a look at some of the people that played a role in developing the first antimicrobial drugs that were used by us. So the first guy that we'll take a look at is a German scientist named Paul Ehrlich. He's actually credited with discovering and developing the first selectively toxic antimicrobial drug that was used by a lot of people. By training, though, he wasn't in the business of finding antimicrobial drugs. Instead, he was in the business of viewing cells under the microscope. And in particular, he tried to find molecules that were really good at staining cells so that they could be viewed under the microscope. So for example, if you took cell biology, you might recognize that the cell you're taking a look at here is a human cheek cell. We can see it under the microscope, or at least we can see it easier because it's been stained with something called methylene blue. It turns out that Paul Ehrlich also worked with methylene blue. He noticed that it could get inside and stain a microorganism called Plasmodium, which is responsible, or one of the organisms responsible for causing malaria. So the thought occurred to him, I have this molecule here called methylene blue that can get inside of these microorganisms called Plasmodium. I wonder if they are toxic to those microorganisms after they get inside. And so he investigated that, and it turned out that methylene blue was indeed toxic to Plasmodium. It occurred to him then that maybe we could use methylene blue to treat people with malaria. The downside, though, is that methylene blue also happened to be toxic to our own cells. So if you added methylene blue to some of our live cells, it ended up killing them. So that's not a good option for a treatment for malaria. But it got Paul Ehrlich thinking, I wonder if there are any molecules out there that could not only get into pathogens, not only kill them, but at the same time leave our own cells alone. So he went on a trek of discovery, basically. So he basically systematically tested hundreds of different compounds to see if they had this property of being selectively toxic only for a pathogen, but not for our cells. And he discovered that the 606th compound that he tested basically was an arsenic derivative, ended up calling it salvarsan. He noticed that it was pretty good at killing off the bacterium that's responsible for causing syphilis, and it was relatively harmless against human cells. And so after he made this discovery, it soon after became widely available, and it was the first, antimicrobial drug that was made available to a lot of people that had selective toxicity. In other words, it was more harmful to the pathogen than it was to our cells. And actually, salvarsan was widely used as the drug of choice to treat syphilis until penicillin came along, which is further down in the story. So to sum this up, we're saying that Paul Ehrlich played a big role in helping to develop the first antimicrobial drug. He was the person. So let's say that he was the first to search for and find a selectively toxic antimicrobial drug that was brought to market. The name of this drug was salvarsan, and it was used to treat syphilis. Again, it was an arsenic derivative. So that sounds kind of hardcore, which means that there were some side effects, but those side effects apparently were tolerable. Right? So people would rather have the side effects associated with salvarsan rather than have syphilis and the consequences of that. One of the other big names in developing antimicrobial drugs was an English microbiologist and physician named Alexander Fleming. Alexander Fleming, by trade, one of his trades was again to work with microbes, specifically Streptococcus, which was a gram-positive. And you can see him in this picture sitting in his lab. I guess the plates tended to stack up in his lab, so it wasn't too much of a neat freak, apparently. But this was good. So this actually led to the chance discovery of penicillin. And so I kind of gave away the punchline, but that's what Alexander Fleming is credited with doing, is discovering penicillin, which was the first true antibiotic that was used by a lot of people. And so here's how he made his discovery. So he went away basically for one weekend. So one of the plates in his lab happened to be left over during that weekend, and when he came back, he noticed this. So again, he worked with Streptococcus, so there are a bunch of Streptococcus colonies growing on the plate. During the weekend, a mold had landed somewhere on the plate. And he noticed that around where this mold had landed, the bacteria had a really, really hard time growing. So check out how faint the bacterial colonies are immediately surrounding where the mold landed. And as you get further away from that mold, the bacterial colonies grew more robustly. They're more thick. And so he saw this and said, oh, it must be that this mold that landed on the plate is secreting something that is slowing down the growth of the bacterial cells. The name of that mold, it was called Penicillium notatum, and so that's why the antibiotic that's derived from it is called penicillin. This happened in about, happened in about 1928-ish when he made this discovery. Previously, that discovery about salvarsan with Paul Ehrlich was made in 1910. So I'm not mentioning the date so that you have to remember, more so that you can get an idea of the sequence of events. So this discovery by Fleming, again, happened in about 1928. He thought it was cool. Maybe I saw some medicinal application for it. You could use it to treat bacterial infections. He wrote it up in a journal. But he had problems isolating the active substance from the mold that was actually killing the bacteria. He couldn't mass produce it. And he noticed that it might be a little bit unstable if you ingest it, right? So if you take in whatever this stuff is that's killing off the bacteria, it breaks down pretty easily in the stomach, he noticed. So he figured it's probably would not be a good medicine if it breaks down easily in the body, right? If it doesn't have enough time to circulate and actually kill the bacteria. So basically, Fleming is credited with discovering this penicillin, but he wasn't able to really make it available to a lot of people. The people who made that happen, so how about if we sum this up? So Alexander Fleming is credited with discovering the first true antibiotic that people use, penicillin. But it wasn't until about 12 to 15 years later, actually during World War II, that two scientists, Howard Florey from England and Ernst Chain from Germany, stumbled across Alexander Fleming's paper describing penicillin. These guys were a little bit better in their biochemistry than Alexander Fleming was, and so they were able to find a way to purify that active substance in the penicillium mold. Not only that, but they were able to mass produce it in a stable way, right? So they're able to produce a lot of it. Not only that, but produce it so that substance that they were making was stable within the human body so that it could be used as a medicine. Right, so here's a picture of the mass production of penicillin that resulted from the techniques that were devised by Howard Florey and Ernst Chain. We mentioned that this happened during World War II, so this was around 1940, and it actually played a big role in determining the outcome of World War II. So because there were these antibiotics that were available for the Allies to use to treat their soldiers with bacterial infections, it kind of gave them a leg up. It's a big, for many reasons, the discovery of penicillin had a lot of historical importance. So how about if we sum this up by saying this, Florey and Chain, what they are credited with doing is that they developed a method for isolating the active compound in penicillin. And again, it played a big role in many different ways in our history. So now that we've taken a look at some of the pioneers in antimicrobial drug discovery, now we'll actually take a look at some examples of antimicrobial drugs and how they work. So the first general category of antimicrobial that we'll take a look at are these things called antibiotics. And when you're talking about antibiotics, those are things that work specifically against bacteria. So antibiotics either inhibit the growth of bacteria, or some of them actually kill the bacterial cells. And typically when you're talking about antibiotics, these are things that are taken internally as medications. There are different ways of categorizing antibiotics generally. And so one way of categorizing antibiotics is according to whether they do this, inhibit the growth of the bacteria, or whether they actually kill the bacteria. So antibiotics that inhibit the growth of bacteria are referred to as bacteriostatic antibiotics. And so static means no movement, and so that's why it's called bacteriostatic, because these are antibiotics that cause no more movement or no more growth of the bacterial cells. On the other hand, if you're talking about antibiotics that actually kill the bacterial cells, those are referred to as bactericidal antibiotics. So that's one way of categorizing antibiotics, is according to whether they're bacteriostatic or bactericidal. Another general way of categorizing antibiotics is according to whether they are broad spectrum or narrow spectrum. And so just like the name implies, whenever you're talking about a broad spectrum antibiotics or antibiotic, it's a drug that works against a big range of different types of bacteria. Typically they're effective against both gram-positives and gram-negatives as well. On the other hand, if you're considering a narrow spectrum antibiotics, those are ones with a more limited scope. Right? So they have effectiveness only against a select group of bacteria. Right? So those are just general ways of categorizing how antibiotics work. Now, finally, we'll get to talking about how specific antibiotics work. So what do these things do? And so we'll take a look at 4 different ways that antibiotics work generally to harm bacteria. So in other words, we're going to take a look at the mechanisms of antibiotic action, how antibiotics work. So the first mechanism that an antibiotic could have is that there are some antibiotics that prevent the bacterial cell wall from being synthesized. And so examples of antibiotics that work in this way include penicillin, the very first antibiotic ever discovered, and also ampicillin, which is considered to be kind of a cousin of penicillin because they have a fairly similar structure if you look at their molecules. And so to see how this works, how these antibiotics work, it'll first help to recall what bacterial cell walls look like. So if you remember from week 1, bacterial cell walls have a general structure like this. It's basically a bunch of sugar chains that are connected to one another via a peptide link. So if you remember from, again, the first week, these circles represent each circle represents an amino acid, and there's a short chain of them that are attached to these sugars called N-acetylmuramic acids. Right, so one of the really important things about this cell wall structure, or one of the really important characteristics if you're the bacterium, is that there's actually a bond that holds together these peptide or these sugar chains. And if you remember that the bonds form between the peptide chains on the sugars. It turns out that there is an enzyme that's responsible for forming that bond. And so we'll let this orange diamond represent that enzyme. The name of that enzyme is called transpeptidase. And if you could visually see what transpeptidase does, again, it quickens the reaction that causes the formation of these peptide bonds that hold together the adjacent sugar chains in the peptidoglycan molecule. So again, transpeptidase is really important because it helps those linkages form between those NAM sugars. Those linkages are really important because without them, the cell wall would fall apart. Right? This provides some strength to the bacterial cell wall. So we mentioned transpeptidase and what it does because we're ultimately trying to figure out how penicillin works. So how this class of antibiotics work. Basically what they do is that they prevent transpeptidase from doing their job. And so if we kind of rewind here, reset, if this is a bacterial cell, and specifically it's the cell's cell wall, and it's in an environment where there are penicillin molecules, what those penicillin molecules do is that they bind to that transpeptidase enzyme and deactivate them. And so if there's no transpeptidase, that means that those linkages between those peptide chains cannot form. And if those bonds cannot form, it means that the bacterial cell has a really weak cell wall that could fall apart. And because it can fall apart, that's what ends up doing the bacteria in. So it's kind of important to note here that if you're considering penicillin, it binds to transpeptidase, which would then prevent these chains from forming. So if you have a cell where the chains have already formed, right, so it's kind of further along in its life where its chains have already formed, penicillin won't do anything against it. Right? So penicillin doesn't break those chains, it only prevents them from forming. So that's how it works, and so this is one example of how antibiotics in general work, is that they prevent cell wall synthesis. And let's say that penicillin specifically, and also ampicillin, the way that they prevent cell wall synthesis is that they deactivate these enzymes, again called transpeptidases. And when that happens, that means that no cross-links can form between these sugar chains. Specifically, they can't form between the short peptides on the NAM sugars. And that really weakens the cell wall of the bacterial cell. All right, so that's one way that some antibiotics work. Here's the second way that other antibiotics work. And what they do to harm bacteria is that they inhibit protein synthesis in those bacteria. And if you remember, proteins are really important because they form all kinds of things in living cells. Right? So there are enzymes, there's proteins that stabilize cell walls, there's proteins that act as messenger molecules. So proteins are a really important thing. So if they are no longer made, that could seriously harm a cell. Some examples of antibiotics that work by preventing protein synthesis in bacteria are tetracycline and something called erythromycin. And as you're looking at this, you're probably thinking, well, wait a minute. How can this be selectively toxic? Right? With that previous thing with inhibiting cell wall synthesis, I can maybe see why that's selectively toxic because it targets and prevents the formation of the cell wall. I can see why that would harm bacteria because bacteria have cell walls, and I can also see why they would not harm our cells because our cells don't have cell walls. This might be a little bit different story, right? So bacteria require proteins. Our cells also require proteins. So again, you might be thinking, well, why is this safe? Is it safe to take a drug that would prevent cells from making our proteins because our cells need to make proteins too? The key to making these types of drugs selectively toxic is the structure of the ribosomes. So you might remember that the ribosome is the place inside of a cell where proteins are produced. And if you take a really close-up look at the ribosomes in prokaryotic cells like bacteria, their structure looks a little bit different than the ribosomes that you find in human cells or eukaryotic cells in general. And so that's the key to these drugs, or to these drugs' selective toxicity, right? So they are able to recognize structures that you only find in prokaryotic organisms. So they recognize prokaryotic ribosome, for example, but they don't recognize eukaryotic ribosomes. And so that's why, for the most part, these cells or these antibiotics don't harm us. So let's sum that up here. So these antibiotics that work by stopping protein synthesis, they are selectively toxic because bacterial ribosomes have a different structure than ours. And so these antibiotics, again, recognize stuff that you find on the prokaryotic ribosome, but not on the eukaryotic ribosome. Another thing about these types of antibiotics is that they tend to have a broad spectrum of activity, so that they kill off a lot of gram-positives and a lot of gram-negatives as well. Which means that if these drugs are taken for a longer period of time, it could actually harm us because we rely on bacteria in order to function in a healthy way. And so you might remember from the first week that one of the benefits of having certain bacteria around is that they might promote certain really healthy things. So, for example, proper metabolism. We'll see next week that being exposed to different microbes might prevent us from getting allergies. So our normal flora, which are really helpful to us, could get wiped out, could get destroyed if you're taking broad-spectrum antibiotics, especially for a long period of time. So that's one of the drawbacks to taking these types of antibiotics. Here's another way that we could get harmed if we took these types of antibiotics for a longish period of time. It turns out that there is a component of our cells that rely directly on bacterial ribosomes in order to work. Right, so here's what I'm talking about. You might remember this from cell biology. So say that this is an animal cell, just a model animal cell. Here are all the organelles inside of that animal cell. You might remember that there's an organelle called the mitochondria that is responsible for producing ATP for the animal cell. The reason why we're highlighting mitochondria here is because those mitochondria actually are bacterial cells that are living inside of our bigger cell. That might sound like a far-out idea, but there's proof for it, right? So if you take a single one of these mitochondria organelles and bust it open and analyze it really closely, you'll find stuff in there that you only find in bacterial cells, right? So there's a membrane structure. If you consider the membrane structure of mitochondria, it's very similar to bacterial cells. It turns out that mitochondria have their own DNA. Not only that, but mitochondria have their own ribosomes. So again, basically, these mitochondria are cells living inside of a bigger cell. And so it's thought that at some point in the past, a bacterial cell, a smaller one, made its way inside of a bigger cell. Those two cells did things that benefited each other, and because of that, that relationship kind of stuck until we have the eukaryotic cells that we have today that still have these mitochondria stuck inside of them. But the point is this: because mitochondria have their own ribosomes, those ribosomes have those are basically bacterial ribosomes, right? So they have a prokaryotic structure, those ribosomes do. And so if you're taking something for especially a long period of time that works by harming prokaryotic ribosomes, our mitochondria could take a hit, because our mitochondria rely directly on prokaryotic ribosomes to carry out their function. So just another thing to be wary of if these drugs are being taken for a longer period of time. And so let's sum that up here. These antibiotics that inhibit protein synthesis, again, have a broad spectrum of activity. This could potentially harm the mitochondria inside of our cells. And the reason why that can occur is because if you take a look at mitochondria, they actually have their own ribosomes. Not only that, but those ribosomes are of the prokaryotic variety. All right, so that's two ways that antibiotics work. We'll take a look at two more. So mechanism number 3, a third way that some types of antibiotics harm bacterial cells is by inhibiting the nucleic acid synthesis of the bacterial cell. And an example of an antibiotic that works in this way is something called quinolones. So quinolones are actually a big class of antibiotic. Quinolones, just a fun fact, are often used to treat urinary tract infections. Similar to protein synthesis, you might be looking at this and thinking, well, wait a second. How can this be selectively toxic? Right? How come taking a drug that inhibits nucleic acid synthesis, how come that does not harm our cells? Because our cells also need to replicate and make nucleic acids. Well, the key is the differences in the way that those nucleic acids are made. So here's a picture of a DNA that's in the process of being replicated. You might remember from earlier this quarter and also from cell biology that several enzymes are needed in order to replicate a DNA molecule. So we talked about DNA polymerase as the enzyme that does the replicating. You might remember from cell biology that there's something called DNA gyrase or topoisomerase that's needed in order to replicate DNA. Basically, the reason why gyrase or topoisomerase is important is because as the DNA becomes unwound, there's a lot of stress that's placed in the section that has not been unwound yet. And so to prevent that stress from happening, which might end up damaging the DNA, what gyrase or topoisomerase does is that it actually nicks the DNA in certain spots in order to relieve that stress, that strain, so that the DNA doesn't get damaged and they can get replicated okay. But the point is this: so several enzymes, including DNA polymerase and gyrase, are needed to replicate DNA. It turns out that the structures of those enzymes are different in prokaryotes as compared to eukaryotes. And so these antibiotics that inhibit nucleic acid synthesis, they recognize only the prokaryotic enzymes, but they don't bind to, generally, the eukaryotic DNA replicating enzymes. So let's say that here. So this class of antimicrobial drug, or antibiotic in this case, is selectively toxic because the DNA replicating enzymes, for example, topoisomerase or gyrase, those are slightly different in eukaryotic cells than in prokaryotic cells. All right, so what we'll do next is take a look at the fourth, and in our case, the last, mechanism of antibiotic action. So it turns out that some antibiotics work by being these things called growth factor analogs. And an example of an antibiotic that is categorized as a growth factor analog is something called sulfonylamides. And I believe sulfonylamides are also commonly used to treat urinary tract infections. So before we take a look at how growth factor analogs work, how about if we define a growth factor analog? Basically, whenever you see this term analog, that means that it's something that is similar to something else. So when you're talking about a growth factor analog, basically it's a molecule that has a really similar structure to a growth factor or to a nutrient that bacterial cells need. So you might be wondering, well, how? Right, so say that a bacterial cell needs nutrient A in order to survive. How can having a molecule that looks a lot like nutrient A why would that be harmful for a bacterial cell? And so to do that, we'll show you an example of a growth factor analog, a sulfanilamide, and we'll show you an example of how it works. And in doing so, hopefully that'll make or help make sense why something called a growth factor analog can be harmful to bacteria. So here we go. Here is an example of a nutrient that bacterial cells require. It's something called para-aminobenzoic acid. The reason why bacteria need it in order to survive is because they actually convert this nutrient into something called folic acid. Folic acid is important because it's used to make nucleotides, and nucleotides are important because they're used to make nucleic acids like DNA and RNA. Right, so ultimately, para-aminobenzoic acid is needed because down the line, it's responsible for making DNA and RNA. It's a really important nutrient that bacteria need in order to grow. So in order for it to be converted ultimately to DNA and RNA, here's what has to happen. The bacteria have to first convert it, like we mentioned, to folic acid. Turns out that bacterial cells have an enzyme that do this converting. So they have enzymes that can recognize and bind to this nutrient molecule and convert it to folic acid. And again, the reason why bacteria would be interested in doing that is because they actually combine folic acid with some other elements in order to produce nucleotides, which are then used to make DNA and RNA. So again, a really important nutrient, para-aminobenzoic acid. So that's all the setup for this. So now we're going to talk about how this class of antimicrobial drugs called growth factor analogs work. And specifically, we're going to take a look at sulfanilamides as an example of a growth factor analog and how it works. So say that here is a sulfanilamide molecule that it has now made its way inside of a bacterial cell. It turns out that that can wreak havoc on a bacterial cell, and you might be able to guess why, based on how this is drawn. Again, this is considered to be a growth factor analog, which means that it has a very similar structure to a nutrient that bacteria need, in this case, para-aminobenzoic acid, or PABA. Because their structures are very similar, that means that this molecule, the sulfanilamide, could just as easily bind to the active site of that enzyme, which would then prevent para-aminobenzoic acid from binding to that enzyme. If that binding doesn't happen, that means that this never gets converted to folic acid, which means that you just threw a wrench into the making of DNA and RNA inside of that bacterial cell. So that's no good for the bacterial cell. It ends up dying eventually. So that's basically how these work. So again, this, by plugging up that active site, the growth factor analog prevents folic acid from being produced, which means that no nucleotides end up being produced. So let's say a little bit more about that then. So we'll do this, put that aside here so you can still see it, and sum it up by saying this. Growth factor analogs, they're again a molecule with a very similar structure to a nutrient molecule. The reason why that would be harmful to a bacterial cell is because it prevents the nutrient, in this case para-aminobenzoic acid, from being metabolized. And the reason why that happens is because the growth factor analog, in this case sulfanilamide, plugs up the enzyme so that it cannot bind to the actual nutrient and convert it. Again, you might be looking at this though, you might be thinking, oh, that's all well and good, but how come this does not harm our cells, right? Because our cells also need nucleotides. So if you're going to take something that prevents nucleotides from being made, why doesn't that harm us? It all has to do, so the selective toxicity of this drug all has to do with how we get this, how we get folic acid. So we've seen that in bacteria. Bacteria get folic acid by taking in this nutrient called PABA and converting it to folic acid. So by having this sulfanilamide in the bacterial cell, that basically throws a wrench in this first step here in that process. So no folic acid ends up being produced. We get our folic acid in a different way, right? So we don't require PABA. We don't have to eat this in order to convert it to folic acid. We actually eat folic acid directly in our diet. So it's actually a form of vitamin B. So as long as you're getting enough vitamin B, that is folic acid, which can then be used to make nucleotides. So even if you were taking this thing called sulfanilamides, it would not prevent us from making folic acid because we just eat folic acid directly from our diet. So let's say that is the last thing here. The reason why these growth factor analogs don't harm us for the most part is because we don't get our folic acid from para-aminobenzoic acid like bacteria do. Instead, we eat our folic acid directly from our diet in the form of vitamin B. And so I forgot to write acid. So we eat our folic acid. It should say directly as vitamin B. So that's basically some of the general mechanisms of antibiotic action. So that's how antibiotics work to harm bacterial cells, but at the same time not harm our cells. What we'll take a peek at next is kind of something that's problematic for us, but a good thing for bacteria. So we'll take a look at ways that bacteria can be resistant to the antibiotics that we just talked about. So previously in class, we've talked about ways that bacteria might acquire the genes that allow them to become antibiotic resistant, right? So we mentioned that mutations could allow bacteria to acquire these genes. Bacteria could also acquire them from other bacteria. So for example, through conjugation or maybe transformation. What we're going to do here is talk about, well, what do those genes allow the bacteria to do specifically? So say that a bacterial cell acquires the gene, either through mutation or through conjugation, acquires that gene that allows them to become resistant to an antibiotic. What is it that that gene encodes for that actually allows them to not be harmed by that antibiotic? So we're going to take a look at 4 of those mechanisms of antibiotic resistance. The first mechanism basically is that there are some bacterial cells that are not harmed by an antibiotic simply because those antibiotics can't penetrate the outer structure of the bacterial cell. So you might remember from way back in the beginning of the quarter that what you're taking a look at here is a schematic of a gram-negative bacterial cell wall. One of the main ways that molecules can enter this outer membrane, one of the ways that molecules can pass through this outer membrane of gram-negative bacteria is by way of these porins. Basically, these are pores, again, that let molecules through. It turns out that the porins have a defined size, and there are certain antibiotics that are just physically too big to be able to go through those porins, and that's what ends up protecting the bacterium, this gram-negative bacterium in this case, against the effects of that antibiotic. All right, so we're taking a look at what makes some bacteria resistant to antibiotics. One of the things that makes bacteria resistant is that they have an outer structure that prevents the antibiotic from entering the bacterial cell. Other things that might prevent that is if there's perhaps a mutation that occurs to some of the outer membrane structure of bacteria. That could also prevent antibiotics from entering the bacterial cell. So again, an example of an outer membrane, or an example of where this is the case, where the outer membrane just does not allow the antibiotic through, is in gram-negatives. And it turns out that they are resistant to certain classes of antibiotics simply because they're just too big to be able to fit through the porins of that gram-negative cell. Another way that bacteria are resistant to the effects of antibiotics is by undergoing a mutation or acquiring a new gene that causes the structure that the antibiotic targets to become a little bit different. Right? So if you remember just a few minutes back, we talked about that there's some antibiotics that work by targeting the ribosomes that you find in bacterial cells. Again, ribosomes are where the bacterium's proteins are produced. In order for those antibiotics to work, they first have to bind to the bacterial ribosome, and after that binding happens, that's what causes the ribosome to not function correctly anymore, so that no proteins are produced in that bacterial cell, which then ends up harming that bacterial cell. So one of the ways that bacteria can become resistant to an antibiotic that works in this way is by undergoing a mutation or acquiring a gene that allows for this target structure, in this case the ribosome, to have a different shape, kind of like this. And so if a mutation like that were to occur, that same antibiotic wouldn't be able to bind to it anymore because simply it has a different shape. And if it doesn't bind to that target structure, that antibiotic is not able to affect how that ribosome works, which means that the ribosome keeps doing what it normally does, which is to make proteins, which means that the bacterium doesn't really feel the effects of it. So another mechanism of antibiotic resistance is that the structure that's targeted by the antibiotic becomes changed, either through mutation or through acquiring genes that changes that structure. And so it can no longer be bound by the antibiotic that otherwise would be able to bind to it. Here is a third mechanism of antibiotic resistance. So some bacteria are resistant to antibiotics because they acquire a gene either through mutation or through gene exchange that encodes for something called an efflux pump. And you might remember from other cells that pumps are things in the membrane that allow things to be taken out of the cell, and energy is required in order to make this happen. So basically, this is how it works. Whenever bacterium, if a bacterium has this ability to make this efflux pump, the reason why that's a good thing for the bacterial cell is because if it ever takes in, or if any antibiotics ever make their way inside of that bacterial cell, this efflux pump allows for that same antibiotic molecule to be kicked right back out so that it doesn't harm the bacterium. So just a third way that some bacteria are resistant to antibiotics is that they have the ability to pump out the antibiotic before that antibiotic can do damage inside of the cell. A fourth way that some bacteria are resistant to antibiotics is by way of this. So it turns out that some bacteria acquire the ability to do this, again, either through mutation or through gene exchange. They acquire a gene that allows them to produce an enzyme that they end up secreting. What those enzymes do is that they deactivate certain antibiotics. So say that you have this bacterial cell that now has this ability to secrete enzymes. That means that if there are any antibiotics, or at least any of a particular type of antibiotic in its environment, those enzymes that it secreted will deactivate that antibiotic, which means that that antibiotic won't harm the bacterial cell. Right? So it's just a fourth way that some bacteria are resistant to antibiotics. They can release an enzyme that actually breaks down or degrades those antibiotic molecules. So in class, we'll talk a little bit more about antibiotic resistance, but these are the basic mechanisms of how bacteria can become resistant or can be resistant to antibiotics. What we'll do next is we'll kind of switch fields. So we've been focusing in on antibiotics, which are molecules that are harmful specifically towards bacteria. It turns out that that there are viral infections also that hurt people, and there are treatments for those viral infections. And so what we'll do next is take a look at some of those antiviral drug treatments. So now we'll take a look at medications that are used to treat viral infections. If you compare all of the antiviral drugs that are available to all of the antibacterial drugs that are out there and available, it turns out that there are a lot more antibacterial drugs. It's relatively hard to make antiviral drugs that are selectively toxic. And the reason why is because if you consider the way that viruses infect cells, so in order for a virus to replicate, like we mentioned earlier this quarter, it absolutely has to get inside of a host cell, take over its machinery so that that host cell then starts producing viral parts instead of human parts. And so because of the way viruses work, right, they actually take over the machinery of our own cells. They use our own cells' ribosomes, they use the nucleotides inside of our own cells. It's hard to find something that'll harm just the virus, but not harm our cells. Right, so that's why it's really tough to come up with a selectively toxic antiviral, because it's hard to prevent viral infection without harming our own cells. But there are some antivirals out there and available, and so what we'll do here is take a look at two examples, or two classes of antiviral drugs. The first one we'll take a look at is something called a nucleoside analog. And so we've mentioned before that what this term analog means is that it's just something that has a very similar structure to something else. So whenever you're talking about a nucleoside analog, that is a molecule that has a really similar structure to a nucleotide. And so basically here's how nucleotide analogs work. So say that here is DNA that is found in a virus. So once a virus infects a cell, it's going to want to copy that DNA. The way that happens is by separating the two strands of DNA. Once they're separated, then DNA polymerase reads those two template strands to insert the complementary nucleotide opposite of each one until you have a completely new molecule built. Nucleoside analogs harm viruses because their structure, again, is very similar to nucleotides. Because of that similarity, that means that a nucleotide analog or nucleoside analog could become inserted into a DNA strand that's in the process of being made. So nucleotide analogs are similar enough in structure to a real nucleotide that it can become inserted into a DNA chain. But there is a difference, though. So it's not completely identical to a real nucleotide. There are some differences. One of those differences prevents new nucleotides from becoming attached after it. So that means that wherever this nucleotide or nucleoside analog becomes inserted into that chain, no matter how hard DNA polymerase tries, it's not going to be able to insert any more nucleotides into that chain after that point. And so because this DNA cannot be fully made, it ends up being degraded. The virus can't make any DNA, which means that the virus can't make any more new viruses, and that's what basically stops the infection. So let's say this, nucleoside analogs, again, they're one class of antiviral drug. Basically, the way that they work is by becoming inserted into a growing viral nucleic acid chain that then prevents the attachment of new nucleotides so that a complete DNA molecule cannot be produced. So what we'll do next is take a look more closely at how one example of a nucleoside analog works. In order to be able to understand, though, how this particular antiviral drug works, it'll first help to know or to review how DNA is synthesized. So here is, say that here is a DNA strand. It's already become single-stranded in preparation for becoming replicated. You know that there's an enzyme called DNA polymerase that helps the complementary nucleotides attach to that template strand. And so you might remember that the complement to T, if you're building a DNA, is A, adenine, and the complement to C is G, or guanine. But here's kind of a key point that you might remember from cell biology. All of the nucleotides that are incoming, right, all the nucleotides that are going to be used to build this new strand, at first, when they come in, they have not one phosphate group, but actually 3 phosphate groups. And here's the reason why. So in order for the new DNA molecule that's built to stay together, right, this sugar and this phosphate have to be able to bind to one another. So if there's no binding, if there's no pasting that happens between this sugar and this phosphate, the molecule will end up falling apart when it's done being built. So it's important to form a covalent bond between these two things in order to have a solid structure. It turns out that energy is required in order to form the covalent bond between the sugar and that phosphate. That energy comes from the breaking off of a couple of these phosphate groups. So it turns out that a lot of energy is released whenever 2 of those phosphates are removed. That's the energy that is used then to form a covalent bond between this sugar and that phosphate so that it stays together after the DNA molecule is built. So kind of a key concept there. So as the DNA polymerase goes down the chain, it'll help the complement to a G attach, which is a C. And again, the incoming nucleotides at first actually have 3 phosphate groups on them. That provides energy to form that covalent bond between the sugar and the phosphate. What's complementary to an A is a T. DNA polymerase will help that thymine become attached. We're going to add in one more detail, though, here. So here's thymine. We're saying that in order for it to become attached here by DNA polymerase, the incoming thymine actually has to have 3 phosphate groups. It turns out that there is another enzyme that's responsible for adding 2 additional phosphate groups to the already existing one. The name of that enzyme is something called thymidine kinase, which we'll abbreviate by saying TK. You might remember from cell biology that generally whenever you're talking about an enzyme that's called a kinase, it's an enzyme that transfers phosphate groups. So in this case, it's going to take phosphate groups and then place them onto this nucleotide, thymine. And after those 2 additional nucleotides have been placed, this thymine now is good to go. It can now be inserted into the growing chain because energy is being provided to form this bond between the sugar and that phosphate group. Right? So just some important review stuff to know about DNA replication, and it'll help to know this before we understand how this example of a nucleoside analog works to prevent viruses from replicating themselves. Right, so let's say this, the take-home point is that there are these enzymes called thymidine kinase, TK for short, that ends up adding phosphate groups to thymidine. This then allows those T's to become inserted into a growing nucleic acid chain. So kind of an important thing. If you consider then, so one more point to finish this thought off actually. So this then allows new DNA to be built, right? If new nucleotides can be inserted into the growing chain. Thymidine kinase, again, is an important enzyme not only to viruses, but to our cells too. So our cells need thymidine kinase. in order to attach two phosphate groups to T's so that they can be attached to DNA so that completely new DNA molecules can be built. So if you consider one of our healthy cells, so here's its nucleus, here is a chromosome, a piece of DNA within that nucleus. What we're saying is that in healthy cells, thymidine kinase would be expected to be found inside, right? Because they're important, again, for the building of new DNA molecules. It turns out that cells that are infected with viruses would also have thymidine kinase, but they could have a lot more thymidine kinase than a healthy cell, a lot more. The reason why is because there are some viral infections that cause the host cell to make a lot more thymidine kinases. And from the virus's point of view, that would be a good thing, because if there's a lot more thymidine kinases, that would allow it to make a lot more nucleic acid at a faster clip so that it could replicate itself in a relatively short period of time. Right, so the point here is this: there are some viruses that cause their host cell, after they infect that host cell, to produce a higher level of thymidine kinase as compared to a normal cell. And the reason why a virus might be interested in doing this is because it allows for more of those viral nucleic acids to be made. Again, because thymidine kinase is needed to replicate nucleic acids. Right? And so this is the reason why this nucleoside analog that we're going to take a look at has selective toxicity. So here we go. How about if we take a look at that example? So again, we're taking a look at how this class of antivirals called nucleoside analogs work. Right now we're going to take a look at a specific nucleoside analog drug called acyclovir, and we'll go over how acyclovir prevents viruses from being replicated, or at least slows down viral replication. All right, so again, say that here is a DNA molecule, viral DNA molecule that's in the process of being replicated. We're going to show you how this drug called acyclovir throws a wrench into this whole process. Acyclovir is something called a nucleoside analog, which means that its structure is very similar to a nucleotide. So specifically, acyclovir's structure is very similar to the T nucleotide. So here it is. Here's acyclovir, very similar structure to T, except there's one big difference. And the difference can be found at the 3' carbon. So real T's, real thymines, have an OH group, it turns out right here. But if you consider a molecule of acyclovir, they are missing an OH group right there. So that's kind of a key role and a key thing as to why acyclovir prevents DNA from being replicated. Right? So if there is thymidine kinase inside of that cell, and again, there's a lot more thymidine kinase in cells infected with viruses as compared to healthy cells, that thymidine kinase will place 2 additional phosphates on that acyclovir molecule. That would then allow that acyclovir molecule to become inserted into the virus's growing DNA. Those 2 additional phosphates will allow this phosphate to stick to that sugar group. Because there is no OH group on the 3' carbon, that makes it hard for any more incoming new nucleotides to become attached to it. So you get what's called chain termination at that point. So no more of that nucleic acid chain is produced. And so basically that's how acyclovir, which is again a nucleoside analog, slows down viral replication by throwing a wrench in the whole process of replicating its DNA. So let's sum that up. We're saying that acyclovir has additional phosphates. So in order for acyclovir to work, it has to have 3 phosphate groups on it, right? So that it can become inserted into the growing viral DNA. Those additional phosphate groups are added to it by this enzyme called thymidine kinase. Once those additional phosphate groups are added on, that acyclovir can then be inserted into the growing nucleic acid chain. So again, throws a wrench into the whole DNA replicating process. So to finish up that thought, that prevents additional nucleotides from being added. You might be wondering how come this, this sounds pretty deadly. You're taking something that prevents DNA synthesis. We just said a few minutes ago, that our cells they normally have thymidine kinase. So wouldn't it be possible for our own cells, because they normally have thymidine kinase, to be harmed by this drug, right? Because that drug can just as easily be inserted into the DNA of a healthy cell as compared to a virus-infected cell. So it turns out that there is some there is danger of healthy cells being harmed by this drug. So if you take a look at the package for acyclovir, there's probably a whole bunch of side effects that are possible because our cells could get harmed by it. The thing is, though, that cells that are infected with viruses are just more likely to be harmed by this drug. And again, the reason why is because if you consider one of our cells that's infected with the virus, there's a lot more thymidine kinase in that cell as compared to one of our healthy cells. Again, thymidine kinase is the enzyme that allows this drug, acyclovir, to become inserted into the growing chain. So basically, thymidine kinase is what activates acyclovir. So because there's a lot more of the enzyme in a virus-infected cell, there's just a lot more, there's just a higher probability that acyclovir, that drug, would become activated in this particular cell. So the point is this: acyclovir has the reason why it's selectively toxic is because it has a greater chance of becoming activated in an infected cell. The reason why is because virus-infected cells, at least some viruses, cause their host cell to contain more thymidine kinase. All right, and so the last thing we'll do is take a look at a second class of antiviral drug. That type of antiviral drug is something called a protease inhibitor. So in order to make sense of it, it'll first help to know what a protease is. Basically, and we might have mentioned this before, a protease is an enzyme that cuts proteins, and it's something that's really important for viral proteins to be made. So it's a really important thing for viruses. Here's the reason why. So say here's one of our cells that's infected with the virus. After getting inside of our cell, that virus is going to try to replicate itself by replicating a lot of its genetic material inside of the nucleus of the host cell. Those pieces of nucleic acid then can be used as instruction manuals in order to produce proteins out at the ribosome. So basically, all of these components then are put together in order to build a new complete viruses. It turns out, though, that the proteins that are produced, a lot of them, when they're first made, aren't in the ready-to-go form, right? Aren't in the active form that the virus is going to use in order to, or the virus is going to need in order to become, an infectious virus. In order to activate a lot of the proteins that are just produced, proteases are required. So green circle with the P means protease. The reason why those are important to viruses, and actually also to our cells, is because they allow for the cleaving of proteins right after they're produced. And the reason why that's important is because it might help with folding of that protein so that it folds in the proper functional way. And so now after you've had the proteins folded in the correct manner, now you can have everything put together to form active viruses that can then infect other cells. So proteases are important because they basically help to produce the final protein product. Right? They're enzymes that cleave immature proteins so that they allow those proteins to become properly folded, which then allows them to have their proper function. So an important thing, proteases. The way that this drug called a protease inhibitor works, the way that they harm viruses is that they prevent proteases from doing their thing. Right? So if you take a protease inhibitor, they bind to those protease enzymes to deactivate them, which means that these proteins that the virus produce can't be cut, which means that they can't be folded properly, which means that no functioning viral proteins are made, which is then not a good thing for the virus, right? So that no new functional viruses are made that can then infect other cells. So that's the skinny then on antimicrobial drugs. We've taken a look at some of the pioneers of antimicrobial drugs. We've taken a look at how antibiotics work. We've taken a look at how bacteria can become resistant to antibiotics. And lastly, we've taken a look at 2 examples of antiviral drugs.